It's recording now. So, this is the second stage of glycolysis or cellular respiration. That's after glycolysis. The moment glucose has been broken down into pyruvate. Now, what happens next to those pyruvate molecules that have been produced? And if we can remember, in glycolysis, we said they can be the pyruvate is broken down into glucose is broken down into two pyruvate compounds, okay, which consists of three carbon atoms. Each pyruvate molecule has a three carbon atom. So those two pyruvate molecules, once they have been produced from glycolysis.
Sorry, sir.
Sorry, sir.
You've stopped sharing. Or it's
I'm
maybe
not
just
sharing.
my network. Oh, okay.
I'm not sharing.
All right.
Sir, there are some people that want to join in.
There are some people that wants to join in. Okay.
Yes.
Okay, let me just. It's good we've not moved. We are just introducing the. There's no auto joining where people can just be joining. I don't know how to do it. Arik.
It's there, but it's the way you generate the link.
Oh, so the way I created it.
Yes.
I just have to be. Okay, no problem. Just keep on reminding me so that we can add as many as we can.
All right.
All right, thank you. Okay. You are able to see the slides, everyone, including those who have just joined?
Yes.
Yes, sir.
Okay. So for those who've just joined, we are looking at the citric acid cycle, which is also called the Krebs cycle. That's in today's lecture. So the citric acid cycle, it's also called the tricarboxylic acid cycle, which is the TCA cycle, and it can also be called the Krebs cycle and also the citric acid cycle. These are the names that you can use to describe this cycle. And I said the citric acid cycle, this is the second stage of cellular respiration. Okay, that's the second stage of cellular respiration. From glycolysis, we go to the citric acid cycle. This is now the second stage of cellular respiration, where the cells are able to produce energy in form of ATP. So from glycolysis, when we have our product, which is pyruvate, it goes further now. It enters into the citric acid cycle, and this is where now there will be also some more cellular respiration and more production of ATP, including NAD and also the FAD-H, which are electron carrier. We will look at those molecules when we look at the electron transport chain, the NADH and the FAD-H, because those two are very crucial when it comes to the transfer of electrons. So the citric acid cycle, I said it's the second stage of cellular respiration, and it has three stages. It's a three-stage process, okay? And it takes place in living cells, okay? It takes place in living cells. So in this process, the organic fuel molecules will be broken down, and there must be a presence of oxygen, okay, in the breaking down of these organic fuels. Then eventually our cells will be able to provide energy, okay, in the form of ATP. So this metabolic process usually it occurs in most plants, animals, fungi, including some bacteria. Many bacteria also, the citric acid cycle, it's able to take place. So in all organisms except in bacteria, the TCA cycle takes place or is carried out in the matrix of the intracellular structures, which is called the mitochondria. Okay, so when you look at glycolysis, if you remember, glycolysis takes place in the cytosol, okay, in the cytoplasm of the cell, okay. That's where glycolysis takes place. But the citric acid cycle in most organisms except bacteria, it takes place in the mitochondria, okay, in the intracellular uh, organelles which are called the mitochondria. We know the mitochondria, that's the power energy of the cell, okay, the power house of the cell, that's the mitochondria. This is where the TTA cycle takes place in most organisms except in bacteria. So this cycle, which is the citric acid cycle, is a series of reaction, and there will be a production of carbon dioxide, two molecules of carbon dioxide, what will be produced in this cycle. Then there will be one molecule of GTP and one molecule of ATP that will also be produced. Okay. Then there will be reduced forms of NADH. Okay. Actually, NADH, three molecules of NADH. Three molecules of NADH will be produced, and one molecule of fat H2 will also be produced. Okay, three molecules of this will be produced, NADH, and also one molecule of fat H will be produced. These are the things that we are going to look at in today's lecture. So almost all the enzymes which are involved in the citric acid cycle, they are soluble enzymes, okay, except just one enzyme, which is the succinate dehydrogenase. Okay, the succinate dehydrogenase, it's one enzyme which is insoluble because it's embedded in the inner membrane of the mitochondrion. Okay, it's embedded in the inner membrane of the mitochondria. That's where you find the succinate dehydrogenase. So this one is different from the other enzymes because these other enzymes, they are soluble, but this one, it's insoluble and it's embedded in the inner membrane. Okay, that's the exception. We will look at this enzyme as we go on. It's usually at the end. At the end, I think at number 6. That's where we'll find this enzyme, somewhere there. So unlike glycolysis, the citric acid cycle, this is a closed loop process. Okay, the last part of the pathway regenerates the compound, which is also now used in the first reaction. Okay, so a closed loop. What will be produced at the end of the citric acid cycle? The end product, which is the malate. Okay, malate will be produced at the end. Okay, that will be at stage number 8. The last stage in the citric acid cycle is step number 8. So whatever will be produced at step number 8 will be recycled and get used at step number 1 again. Okay, so the malate which will be produced at step number 8 will be reused, okay, at step number 1, and the process starts all over again. That's why it said this is a closed loop cycle. Okay, you understand when we look at the whole process from the first enzyme to the last enzyme. So what will happen is the end product, which is malate, the moment it's produced, it will again be converted to the first molecule. And the first molecule, that's oxaloacetate. Okay, that's where the malate will be converted, and the oxaloacetate we will start again, combine with the acetyl CoA and produce citrate, and the process restarts. So we will look at these things as we go on. So each turn of the cycle forms one GTP. So one GTP is formed at each turn of the cycle, and also one ATP is also produced, as well as three NAD molecules. Okay, these are the reduced forms of NAD. Three molecules are produced, including one reduced fat H2 molecule. And these two, the NAD, let me just change the pointer. Right. Okay. So these two, the NAD H, and also the FAD H2. These are the two molecules which are very crucial and very important. Okay, because these are reduced forms. They are in reduced forms, and they are electron carriers. Okay, they are able to carry electrons and go on and donate them onto another process or another reaction. So these two will go on and take part in the further steps of cellular respiration. Okay, these two, the NAD H and the FAD H, will go on and take place in the other steps of cellular respiration to produce more ATP again for the same cells. Okay, that's why these two are very, very important molecules. So this is considered an aerobic pathway because the two molecules, the NAD and the NADH, that are produced, these must transfer their electrons to the next pathway in the system. That next pathway now in the system, that next pathway where these two will transfer their electrons, that is now the pathway that will require the use of oxygen. Okay, so when we are done with this pathway, we will look at the electron transport chain. Okay, we will look at the electron transport chain, and you'll be able to understand more concerning the NADH and the FAD H2. Okay. So if the transfer of electrons does not occur, the oxidation steps of the citric acid cycle can also not take place. Okay, so there should be a transfer, and the transfer, that's a transfer of electrons, and that's why we have those two electron carriers, the NAD and the FAD. They are able to accept the electrons, and once they accept those electrons, then the TCA cycle can take place. And note that the citric acid cycle produces very little ATP, okay, and does not directly consume oxygen because it's the next step that will consume oxygen. The citric acid cycle does not directly consume oxygen, but the next step that we use the NADH and the FAD are H2. That is the step now that we will directly use oxygen. Okay, that's what this point is trying to explain here. So we have the 4-carbon molecule. So the 4-carbon molecule that we have, which starts the citric acid cycle, is what is called the oxaloacetate. Okay, this is the 4-carbon molecule that we have. So this molecule began the cycle, and this is where that began the cycle is regenerated after the 8 steps of the citric acid cycle. This one, it starts the cycle, and after the cycle, after the citric acid cycle has completed, at the end of the full cycle, the malate, again, the end product of the citric acid cycle, will eventually be converted into the same oxaloacetate. Okay, that's what it means here. From the first product, which is the oxaloacetate, it starts the citric acid cycle. Once the cycle completes, at the end product, which is malate, will again be reconverted into oxaloacetate, and the cycle restarts itself. It starts again from oxaloacetate. So the citric acid cycle, it consists of 8 steps. Okay, it has 8 steps. So in these 8 steps, what is involved? There is redox reactions. Okay, we have redox reactions. Okay, reduction oxidation reactions. We have dehydration. Okay, removal of water, then addition of water, which is hydration, and we have decarboxylation reactions. Okay, the removal of carbonic compounds. That's what will be involved in these 8 steps of the citric acid cycle. So if you remember, in glycolysis we have 10 steps. You have to take note of that. But in the citric acid cycle, it's an 8 step process. So let's move. Let's look at the reactions. So the citric acid cycle starts from the breakdown of pyruvate. The pyruvate that comes from glycolysis. This is where now it's broken down, and it's one product now that is being used in the citric acid cycle. So the conversion of pyruvate from glycolysis to acetyl-CoA, this is a 3 step process. Okay, that's from glycolysis to acetyl-CoA. This is a 3 step process, and it involves the loss of a carboxylic group. Okay, the carboxylic group from the pyruvate molecule is going to be lost, and once it's lost, one product which will be produced, it's a carbon dioxide molecule. Okay, that's one molecule that will be produced. So in these reactions, the moment the pyruvate from glycolysis has been produced, then it undergoes these 3 steps. Okay, where it's broken down. So in the breaking down of pyruvate, this is where we have the loss of the carboxylic group, and carbon dioxide is produced. Then we know that pyruvate has 3 carbon atoms. Okay, pyruvate has 3 carbon atoms. So once one carbon atom has been removed and carbon dioxide has been produced, it means the pyruvate molecule now will just remain with 2 carbon atoms. It will remain with 2 carbon atoms. Once these 2 carbon atoms remains, these are now the atoms now that will be transferred to the enzyme CoA. Okay, the enzyme CoA, this is the enzyme that will act on these 2 carbon atoms and produce acetyl-CoA. Okay, this is the molecule that we need, the acetyl-CoA. So there is the enzyme CoA that works on the 2 carbon atoms that remains after the loss of a carboxylic group from the pyruvate, and these two carbon atoms will be transferred to the enzyme CoA, and what will be produced is the acetyl CoA. Okay, that's the molecule that will be produced. So let's move. So the acetyl CoA now, this acetyl CoA combine with oxaloacetate and continue the reaction of the cycle. So the acetyl CoA will combine with oxaloacetate, and there the process continues. Okay, that's what is happening in this cycle. So we have the group which is called the hydroxyethyl group. This is oxidized to an acetyl group. Okay, once it's oxidized to an acetyl group, and the electrons are picked up by what we call the NAD. Okay, this is now the NAD that comes in. So this NAD will be converted to the NADH, which is a reduced form of NAD. And the NADH is what is called the high energy is what consists of the high energy electrons, which will be able to be donated in a later stage, and the cell will be able to generate ATP. Okay, because of these high energy electrons that the NAD has. Those electrons, the moment they are donated, then there will be a generation of ATP. That's what is happening here. Are we together? Are we together?
Yes, sir.
Yes, sir.
Okay, that's good. Okay, so let's move to the next. Okay, so this is our cell. We have the cytosol, which is the cytoplasm, and we have the mitochondrion, which is the mitochondria. Okay, so outside the mitochondria, this is where we have our pyruvate. This is our pyruvate molecule. So this pyruvate, it's coming from glycolysis. So the pyruvate now, it's the product now that is being broken down. Okay, and this process takes place in the cytoplasm. Okay, that's where glycolysis takes place. Glycolysis takes place in the cytoplasm. We need to know that, or we've forgotten about glycolysis. Have we forgotten?
No.
We've not forgotten.
No.
Okay, if we've not forgotten, that's good. But let's not be forgetting. Okay, let's not be forgetting. Just a minute. Just want to send a message someone. Okay. Okay, let's continue. So, so this pyruvate, it's from glycolysis, and it's now the molecule that will be broken down. Okay, so the pyruvate has 3 carbon atoms, 1, 2, 3. So the carboxylic group, this one will be lost. Okay, this one will be lost. Usually, there is an enzyme which I've not mentioned. There is an enzyme that comes in here, the pyruvate carboxylase. That enzyme, it's the one that removes the carboxylic group from this pyruvate. Okay, once the carboxylic group has been removed by pyruvate carboxylase, then this that remains now will be transferred to a coenzyme A. Okay, the coenzyme A. And this carboxylic group will be converted to carbon dioxide. This carbon dioxide, which is here. Okay, it's coming from the carboxylic group. Okay, then the coenzyme A now will get the remaining 2 carbons and convert them to an acetyl-CoA. So this acetyl-CoA, we need it so much because it's what we are going to need. It's going to combine with the oxaloacetate and produce citrate. Okay, that's why we need this acetyl-CoA. Then the acetate molecule, once it combines with the acetyl-CoA, then the citrate will be produced and the process continues. The process continues. That's the citric acid cycle. Let's move. So let's look at the steps now. So the first step is a condensation step. A condensation meaning there is a condensation reaction that is taking place. That's in the first step of the citric acid cycle. So in this first step, there is the combining of the 2 carbon acetyl group molecules, the ones that comes from the acetyl-CoA. Okay, so those 2 carbons, those 2 carbon atom molecules that forms the acetyl-CoA will combine with a 4 carbon atom molecule, which is the oxaloacetate. And what will be formed now is a 6 carbon molecule, which is called citrate. Okay, this is what is happening in the first step. This is what is happening in the first step. There is a condensation reaction that is taking place. So the acetyl-CoA, which was produced, combines with the oxaloacetate. So the acetyl-CoA has 2 carbon atoms, while the oxaloacetate has 4 carbon atoms. And what will be formed is a 6 carbon atom molecule, carbon molecule, which is called the citrate. And this is how citrate is produced. Okay, that's how citrate is produced. So from here now, the CoA, because we know that the acetyl-CoA has the CoA which is attached. Let's go back to the structure. Okay, so when you look at the acetyl-CoA, there is this CoA, this molecule. Okay, that's what is making the acetyl-CoA. This CoA comes from the CoA enzyme. Okay, it combines with the 2 carbon atoms that came from the pyruvate and forms the acetyl-CoA. So this CoA, this functional group, the CoA, this is now what we are trying to talk about here. So the CoA is bound to a sulfhydryl group, which has a sulfur and hydrogen atom. Sulfur and hydrogen atom makes up the sulfhydryl group. Okay, and this is the functional group which is attached to the acetyl-CoA. So this one, the moment the acetyl-CoA combines with the oxaloacetate, this one diffuses away. Okay, the sulfhydryl group diffuses away and to eventually combine with another acetyl group that can also come in and combine with oxaloacetate, and the process continues. So this step is irreversible because it is highly hexagonic. Highly hexagonic, it needs a lot of energy. It uses a lot of energy, but it can't on itself produce any energy, and it's called an hexagonic reaction, and it's irreversible. It can't go back. Okay, that's the removal of this group, the sulfhydryl group. It diffuses away from that molecule and it's gone just like that. And this is an irreversible reaction. So we continue. So this irreversible reaction, the rate of this reaction is controlled by negative feedback and the amount of ATP that is available. Okay, so in case the ATP molecules levels increases, then the rate of this reaction also will decrease. Okay, there is a negative feedback. If the levels of ATP increases, then the levels of the reaction, the rate of this reaction decreases. Okay, it's ATP plays a negative feedback role. So if ATP is in short supply, then the rate also of this irreversible reaction also will increase. That's if the cell has low levels of ATP. This step, it is characterized by an enzyme that is called the citrate synthase. Okay, the citrate synthase. That's the enzyme that is involved in the production of citrate. Okay, the citrate synthase, where the acetyl-CoA combines with the oxaloacetate and sulfhydryl group is removed. Okay, this is the enzyme that is involved. This enzyme, it's the one that is involved. Then we look at the second step. So in the second step, what will happen is a dehydration reaction. The citrate will lose one water molecule and gains another as citrate is converted into its isomer. So what will be produced now is what is called the isocitrate. This is an isomer of citrate. So there is dehydration. After dehydration, there is also an a hydration reaction, and what is produced now is an isomer of citrate, which is called an isocitrate. And the enzyme that characterizes this reaction is what is called the aconitase enzyme. This is the enzyme that is involved in step 2, okay, where citrate loses one water molecule and eventually it gains another one and ends up into an isomer of itself, which is called the isocitrate. And the enzyme that is involved is what is called the aconitase. That's the second stage of the TCA cycle, or the citric acid cycle. So we look at the third stage. So the third stage, now we have our product, which is the isocitrate. So in the third stage, the isocitrate now will be oxidized. Okay, it's going to be oxidized, and the product now that will be produced is a 5-carbon molecule, which is called alpha ketoglutarate. So meaning one carbon has been lost because the isocitrate is a 6-carbon molecule. So for this alpha ketoglutarate to be produced, it means we've lost one carbon molecule has been lost, and this alpha ketoglutarate, it's a 5-carbon molecule. So once that one carbon molecule has been lost, it's going to lead to the production of carbon dioxide. So carbon dioxide is produced, and then there are 2 electrons. Okay, there are also 2 electrons which reduce NAD to NADH. Okay, so we had our first NAD when we just started, and we have another NAD which has been produced through an intermediate product which is called the oxalosuccinate. This one is just an intermediate product, and this reaction is characterized by the enzyme which is called isocitrate dehydrogenase. Okay, that's the enzyme that is involved in step number 3 of the citric acid cycle. So we continue with the third step. So this step, it is also regulated by negative feedback, okay, which is from ATP and also the NAD, from ATP and the NAD. This is the negative feedback that controls the step number 3. So the moment we have high levels of ATP, then this third step, the reaction decreases, okay, because there is a negative feedback. So the moment we have high levels of ATP and NAD, then the reaction decreases. Then there is a positive feedback or positive effect by ADP. Okay, so if we have high levels of ADP, the reaction also increases. Okay, the reaction will increase in case the cell has high levels of ADP. It has a positive effect on this reaction, or step number 3. Then we look at step number 4. So under the step number 4, this is where we have a succinate group, okay, which is the product of step number 4. So the succinate group will be produced in this step, okay. Let's not forget that in step number 3, what we add was the alpha keto glutarate, okay. That's a product that we add. Now at step number 4, we have a succinate group, which is a product of this step number 4. So the CoA binds to the succinyl group to form the succinyl CoA, okay. Then the enzyme that will be involved in this reaction is the enzyme that is called the alpha keto glutarate dehydrogenase. This is the enzyme that comes in, okay, at step number 4. Okay, we have the alpha keto glutarate which comes in, and what will be produced, it's the succinyl CoA, which comes from the succinyl group that attaches itself to a CoA enzyme, and we have what is called the succinyl CoA as the product that is produced at stage number 4. So this enzyme, which is the alpha ketoglutarate dehydrogenase, this one is regulated by a feedback inhibition, okay, a feedback inhibition of ATP, then succinyl CoA, and also the NAD H. These are the 3 molecules that are able to regulate stage number 4. There is a feedback inhibition of ATP. The moment we have a lot of ATP or succinyl CoA or NAD H, then this will inhibit the enzyme. This enzyme, which is alpha keto glutarate dehydrogenase. And once this enzyme has been inhibited, then the alpha keto glutarate will not be converted to succinyl CoA. That's our step number 4. So the steps, step number 3 and step number 4, these are both oxidation and decarboxylation steps, which release electrons that reduce the NAD to NADH, and also the carboxyl group that is lost. It goes on and produces carbon dioxide. Okay, so it's an oxidation reaction and decarboxylation reaction that takes place at stage number 3 and stage number 4. And what will happen is that those electrons which are released at stage number 3 and 4, they reduce NAD to NADH, and also the production of carbon dioxide because of the decarboxylation, the removal of a carbon or one carbon atom from the molecule that we have. Okay, that's what happens here. So we go on now and look at stage number 5 of the tricarboxylic acid cycle. So at stage number 5 now, this is now where we have a phosphate group that comes in. So a phosphate group comes in, is substituted for a coenzyme. So the coenzyme from the succinyl CoA is removed, okay, and we have a phosphate group that comes in. So a high energy bond is formed once the phosphate group joins in. There is a high energy bond that is formed. So this high energy is the energy now that is used as substrate level phosphorylation during the conversion of the succinyl group to succinate. Okay, we look at another step where the succinyl group or the succinyl CoA will be converted to succinate. So at this stage, since the succinyl CoA has been the CoA has been substituted by the phosphate group, what remains only is the succinyl group without the CoA attaching itself to this molecule. And what will be produced now at this stage is either the guanine triphosphate, which is the GTP, or the adenine triphosphate, which is the ATP. That's at step number 5. These are the two things that will be produced, GTP or ATP. Okay, because we know the phosphate group has been attached here, this phosphate group. So let's not get confused where the GTP or the ATP is coming from now, because we have the phosphate group already here, and what will be produced here is either the GTP or the ATP, and the reaction is now catalyzed by the succinyl CoA synthase. This is the enzyme that catalyzes this reaction or this step, which is step number 5, involving an intermediate succinyl phosphate, and the product that will be formed now is the succinate. This is how the succinate comes in. Okay, the succinate is produced at step number 5 of the citric acid cycle. So there are, when you look at the GTP and the ATP, there are two forms of the enzyme, okay, which is this enzyme, the succinyl CoA synthase. So it has two isoforms of itself. So these two enzymes or isoenzymes of the succinyl synthase, this enzyme. So for this step to occur, it depends upon the type of the tissue. Okay, it depends upon the type of the tissue where these cells are. Okay, for example, the brain tissue and the muscle tissues, they need a lot of energy, and you expect more of ATP to be produced as compared to GTP. Okay, but when you go to other tissues like the liver, you expect more of GTP to be produced. So these isoenzymes of this enzyme, which is the succinyl phosphate, will be available depending on the tissue where this process is taking place. So one form of the enzyme is found in the tissues that use large amounts of ATP. An example is the heart, skeletal muscle, where you expect a lot of ATP to be used. Okay, that's one isoform of this succinyl phosphate. That's where you find it, and the more ATP will be produced. Then the other one will be found in the tissues. Okay, in the tissues that have a high number of anabolic pathways. An example is the liver. This form produces GTP. So you have to take note that GTP is energetically equivalent to ATP. However, its use is more restricted. Okay, that's the only difference. Its use is more restricted, although it's energetically equivalent to ATP. Okay, so let's now look at the step number 6. So at step number 6, this step now, we have what is called the fumarate, which is produced at step number 6. So the fumarate comes from the succinate, okay, which was produced at step number 5. So at step number 6, there is a dehydration process that takes place, okay, and this dehydration will convert succinate to fumarate, okay. That's what is happening at step number 6. So there are 2 hydrogen atoms that are transferred to the FAD. Now, instead of the NAD, this is where the FAD comes in, okay? And what will be produced? It's the FAD H2, which will be produced. So here they are saying, what we are saying here is the energy that is contained in the electrons, or in these electrons which are transferred to FADs to produce FAD H of this atom is insufficient. Okay, the energy cannot reduce the NAD. Okay, the energy that is produced here, or that is available here, that reduces FAD to FAD H, it's insufficient to reduce NAD to NAD H, but adequate to reduce FAD to FAD H2. Okay, that's why the FAD comes in. This energy which is involved here at our step number 6, it's insufficient to reduce the NAD to NAD H. That's why the only thing that can be reduced, it's the FAD to FAD H2. Okay, that's what is happening at position number 6. So unlike the NAD, this carrier remains attached to the enzyme and transfers the electrons to the electron transport chain directly. So what they mean here is they mean the FAD H, the FAD H2. It's an electron carrier. So this one will remain attached to the enzyme inside the mitochondria membrane. Okay, and it's going to transfer the electrons directly to the electron transport chain. So when we look at the electron transport chain, you'll be able to know how the FAD H2 and the NAD H transfers these electrons, and in the end leads to aerobic respiration, or beta, or what's the other name I can use? It's aerobic respiration, or what? Cellular respiration. A way in which our cells are able to produce energy using these two, the FAD H2 and the NAD H. So the FAD H2 remains attached to the enzyme in the mitochondria, and it's going to be transferring the electrons onto the electron transport chain. So we'll look at the electron transport chain in details, and you'll be able to see how it also works as another pathway in cellular respiration. So step 7. So at step 7 now, a water molecule is added to fumarate. Okay, that's at step 7. There is a water molecule that is added to fumarate. That's where now hydration comes in. Whenever there is addition of water, that process is the hydration. So this stage is now called the hydration stage because there is an addition of a water molecule, and what is produced at stage number 7 is what is called the malate. Okay, malate is what is produced, and it's the end product of the O process. Okay, it's the end product of the citric acid cycle or the Krebs cycle, whichever name you can use. And the enzyme that is involved at this stage is what is called the fumarate hydratase. Okay, the fumarate hydratase, this is the enzyme that is involved at step number 7 in the addition of a water molecule to the fumarate, and malate is produced. Okay, so we go on now and look at the last stage. So like I said this process is a closed loop process. Our end product, which is malate, it's going to be or it's going to be oxidized again or worked on, and what is regenerated, it's the oxaloacetate. And the oxaloacetate, it was the first thing that we were looking at. Okay, from the breaking down of pyruvate, the acetyl CoA comes in, combines with the oxaloacetate, and produces the citrate. So this whole process, what will be produced at the end, it's the oxaloacetate. The same thing that we started with, the malate will be converted to oxaloacetate, and by oxidizing malate, the enzyme that is involved at this stage is what is called the L-malate dehydrogenase. Okay, that's the enzyme that is involved at stage number 8, and there will be one molecule, also a molecule of NAD, which will be produced through the actions of this enzyme, which is the L-malate dehydrogenase. And that's how now the citric acid cycle operate. This is how this pathway is. We can see that there are a lot of electron carriers, the NAD, that are produced. Okay, about 3 NADH molecules, and also the fat H2 molecule. And these molecules that are produced will enter into the electron transport chain. So like I said, the citric acid cycle produces very little ATP. Okay, just one molecule of ATP or one molecule of GTP, very little, but it produces a lot of NADH. Okay, so that these 3 molecules of NADH and 1 molecule of the fat H2 will enter the electron transport chain, and this is where a lot of ATP will be produced, because that is oxidative respiration, where oxygen is needed. Oxygen needs to be present when we go to the electron transport chain. We'll be able to see that oxygen needs to be present, and this is where the NAD and the NADH and the fat H are very, very important. Okay, so there are lots that are produced in this process in the TCA cycle, the NADH and the fat H2, while just a molecule of ATP is what is produced. So the oxaloacetate, like I said, it goes on and reacts with the acetyl CoA and produce citrate, and the cycle continues. It restarts again. This is the overall picture of what we've been talking about. Okay, so we started from here. This is our CoA, where we started from, attaching itself to the CoA sulfhydryl CoA. Okay, the sulfur that is attached to the CoA enzyme reacts with the molecule which is coming from the pyruvate. Then we have the acetyl CoA, okay, which is produced. the acetyl-CoA, we come in, we have the oxaloacetate. This one combines with acetyl-CoA. A water molecule is used up, okay? Used up. Then we have the sulfhydryl-CoA, which is removed, okay? This one is released, and the citrate is produced. The citrate, we said it's a 6-carbon atom molecule. The citrate will be converted to its isomer, which is the isocitrate. Okay, that's at number 3. Okay, that's at number 3. So the isocitrate goes on, converts the isocitrate to alpha ketoglutarate. Okay, alpha ketoglutarate. From the alpha ketoglutarate, we have the CoA enzyme that comes in again and attaches itself to the alpha ketoglutarate, and what is produced is the sinew CoA molecule. Okay, from here, this is where we have our phosphate group attaching itself to the sinew CoA, and the CoA is removed. Then from here we have the succinate, which is produced. At this stage, this is where we have our GTP or ATP produced. Okay, we can either have the GTP or the ATP produced at this stage. Okay, then from the succinate molecule, now this is now where we have the fumarate, which is formed from the succinate. We have the fumarate that is formed from the succinate. So at this stage, from the succinate to the fumarate, this is where we have the FAD now, the FAD being reduced to fatty H. This is where we are saying the energy which is here at this stage, the energy which is here, it's insufficient. Okay, it's insufficient to reduce the NAD to NADH, but it can reduce the FAD to FADH. Okay, and this one will remain attached to the enzyme and go on and transfer electrons to the electron transport chain. Then the fumarate will be converted to malate. That's after an addition of a water molecule. A water molecule here is added, and the fumarate is converted to malate. Then the malate, like I said, this process is a closed loop process. This malate will eventually be converted to the oxaloacetate. It goes on and gets converted to the oxaloacetate, and there is one NADH that is produced at this stage from the NAD. The oxaloacetate will re-enter the process, and the process begins again. Okay, that's all about this process, which is the tricarboxylic acid cycle or the TCA cycle. This is just the same as the other diagram, but here instead of saying oxalosuccinic or oxalosuccinyl, it's using the acid, the citric acid, but it's the same as what I've talked about from here. Any questions where you have problems? This is where this one is ending. Where do we have problems about the TCA cycle? Where is it not clear?
You can just that to the last diagram, the one which is showing the this same one, where it's showing the GTP, which is reduced to DGP. So you said we can use either ATP.
Yes,
or we
ATP
just
or GTP. Here, what is produced here, it depends on the tissue where it's taking place. So if it's in the liver, it's the GTP that will be produced. Okay. If it's in the liver, it's the GTP that will be produced. But if it's in the muscle cells or the heart, or muscle cells or the brain cells or the heart cells, then it's the ATP, because these cells, they need a lot of energy. Okay, but you can use whichever you want. If it's ATP or GTP, it's all the same. Okay, there's no problem.
So I have a question.
Yes.
Just at the first point, on the same circle, the same diagram.
Okay. Okay.
Yes. So I think I didn't understand on the first step where acetyl CoA. So the first product that we have coming from glycolysis, which is pyruvate.
Yes, that's the same. Yeah, the first product that
Yes.
we have.
So is it going to react with acetyl CoA or for it to
No,
get into the circle?
it's broken down. The acetyl CoA, the pyruvate, it's a 3-carbon molecule. You remember?
Yes.
Yes, it's a 3-carbon molecule. So the pyruvate is going to be decarboxylated. Okay. One carbon molecule, the carboxyl group is removed. Okay. The carboxyl group is removed. Once the carboxyl group is removed, what will remain is a two carbon molecule, okay, which is the acetyl CoA. The 2-carbon molecule that remains from the pyruvate, it reacts with the CoA enzyme. This is an enzyme. You've seen this enzyme?
Yes.
The CoA enzyme. Then this is a 2-carbon molecule. You've seen the carbons are just two. 1, 2. Okay. You've seen the number of carbons here. So
Yes.
these two carbons, they've come from pyruvate. Okay. Pyruvate had 3 carbon molecules. So 1 carbon molecule has been decarboxylated. It has been removed. Do you understand there?
Yes.
Yes, 1 carbon molecule has been removed. And what remains now from the 3-carbon pyruvate is just 2 carbons that remains. So those 2 carbons now, that molecule with 2 carbons, it's now the one that is acted upon by the CoA enzyme. This enzyme, the CoA enzyme. So this enzyme, it acts on this molecule, the 2-carbon molecule that came from pyruvate. And what will be produced now is what is called the CoA, this compound, which is called the CoA. It only has 2 carbons. Okay, that's the CoA. It only has 2 carbons. It's a 2-carbon compound. Now this CoA, the CoA, it's the one that combines with oxaloacetate.
Okay.
Understand there? Yes, it combines with the oxaloacetate and forms the citrate, which we have here, the citrate.
Okay.
This is 4 carbons from oxaloacetate and 2 carbons from acetyl CoA. And this forms 6 carbons compound, which is citrate. Okay. The citrate, it has 6 carbons. Because we add the acetyl CoA has 2 carbons. Okay. Have you seen it?
Yes.
Are you seeing it? It has 2 carbons. Then the oxaloacetate, it has 4 carbons. So what will be produced here is the citrate. So this citrate, it has 6 carbons. 2 from acetyl CoA and 4 from oxaloacetate. You get it?
I get it now.
You get it?
Yes.
Then the sulfhydryl group now, that sulfhydryl group CoA, including the CoA, will be removed. This group is lost. Okay, and we just have now the compound citrate without anything attached here. This group, the CoA group, was the sulfhydryl group which we add. It's lost. That's why we only remain with this citrate now. That's where we have it. Are you okay there?
Yes.
Okay. If you are
Thank
okay,
you.
then that's good. You are most welcome. Any other question concerning the same diagram? Or anything, not just the diagram. Anything from where we started from. From where we started from. Any questions or where you are not clear? Okay. If there are no questions, then this is where we end for today. Mr. Arik, is Arik there?
Yes, sir. I'm here.
Okay. We can end from here today, and next we'll do the electron transport chain. I'm sending you the notes just now
All right.
for the electron transport chain. Yes.
Okay. Okay.
All right. Thank you. I'm sending you the notes just now.
All right, sir.
All right. Thank you so much.
All right. Thank you.
Thank you.
Yeah.